My name is Wang Hongray. I produced a paper submitted to AMETA. The title is Rhizosphere Interface Microbiome Reassembly by Arbuscular mycorrhizal fungi Weakens Cadmium Migration Dynamics. Cadmium cannot be degraded and is easy to be absorbed by crops. Inoculate microbials can solve these problems, and AMF is one of them, even alive in polluted soil. It can enhance plant nutrition and also can reduce the absorption of heavy metals. We found that regulation of plant rhizosphere by AMF can prevent the cadmium uptake, which is also the first line of defense, and the survival of microbial groups is a key hub to achieve it. The effect of AMF on microbiome may lead to variation core functions towards heavy metal resistance, and is related to keystone. In general, we aim to address the following questions. 1. Whether AMF reduces cadmium migration. 2. What roles metabolites play in soil microbiome. 3. How AMF regulates the metabolite-soil microbiome interactions. The next is method. A composite medium was formed by mixing cornfield soil with flower soil and vermiculite. Then added AMF bactericide from 2 treatments and loaded into root box. When the root fraction reached 90%, cadmium were added, and a total of 4 treatments were formed, namely CK, CD, AM, AMCD, continuing 25 days. The first is to fluorescent dye protocol as figures, and the Rhizosphere soil was collected for parameter determination and high throughput sequencing of microbial. The next is results. On the 27, cadmium showed a pattern of distribution along the root, and under 33, a hotspot of cadmium appeared around the root. However, compared with the CD, the hotspots in the AMCD was reduced, and the content of exchangeable cadmium in the AMCD is lower than that in the CD, but organic bound cadmium increases. The next is bacterial community. The classification analysis showed that bacteria were mainly composed of 6 phyla, as shown in figure. Compared with CK, proteobacteria and others was reduced after CD. However, compared with CD, Della and others treated with AMCD were increased, even higher than CK, but Della was not different with CK. Based on the comparison of Bricardies and Anosine, it was found that both the AM and CD resulted in differences in bacteria. CK and AMCD are random for process, while CD and AM are deterministic process, but the former is homogeneous selection and later is variable selection. Left-C analysis, paired t-text, simple analysis showed the bacteria changed under different treatments. By constructing networks to identify as keystone species, we found that AM and CD can infect microbiome by recruiting their preferred keystone species. We predict the functions of bacteria and found that the AMCD increased heavy metal transport function. We named these bacteria as HM remover. A core expression network was used to explore the main factors of microbial change. The results showed that flavonoids are the key hubs affecting community and keystone, and AMF changes microbiome by increasing flavonoid secretion. Using structural equation model, it was found that root secretions was the key signal reduced cadmium migrations at the figures, and the microbial explanation rate was the largest. The study shows that control of the metabolic is essential for interactions with the microbiome. Under the cadmium toxicity, AMF assembles heavy metal resistance microbiome in the rhizosphere, reducing cadmium migration. Therefore, a new scientific explanation was provided for AMF to improve cadmium tolerance. Thank you for your listen.